Sorry, what are you? Shazam, okay? I'm Mariam from Mariam. Mariam from Mariam. Good. So, yeah. Are you all schooling or private candidates? He's a private candidate, wherever you are. All at school, right? Okay. So, what about the term? You're private. Okay. What about the term tests? Do you all do term tests at schools? Which term test have you completed by now? First term. First term. Still first term, eh? I was a private candidate, so I too have no idea regarding the school system, since I was a private candidate as well. So being a private candidate, the only disadvantage is not having term test, but I hope you all write papers and manage to complete the rest of the subjects as well. Right? Yeah. Online students are joining. Online students, can you just let me know whether you all can hear me? Is the chat busy?
I think that's the problem now.
Why are you working during
After should try my
okay. Have they connected the mic? The microphone is connected. Hello, can you all hear me? I'm enable the chat. Right. Mic. Okay. मैं chat को किसे लोग ना तो इन मोबाइल okay. ऐसे दादा मुझे एक मिनट दादा फाइल फाइल को हमें स्कैन करना है what is the value the video say oh we are if online students you all can hear me, can you raise your hand? Yeah, they can hear. Right. So once again, we'll begin the class. So today's class will be a free session. So my plan is to do the class until 11:30. As usual, in between you will get a break. So today we are going to start off with primary growth, right? So we're talking about plants. The primary growth is one of the most important procedures that is happening in plants. So before talking about the primary growth, I hope you have revised about all the cells, all the tissues, the meristems, because all these concepts are required to continue, move forward, because you need to know what is collenchyma, the function of collenchyma, the function of sclerenchyma, the functions of parenchyma, okay, and also you need to know what are meristematic tissues and what are the functions of meristematic tissues. Everything you need to know in order to move to the next section, right? So have you all revised? Have you revised? Huh? Not yet. So the thing is, you can be very friendly with me because, like, you know, I'm just 2 or 3 years older than you. So you can talk to me and yeah, you can be friendly with me. The class should be active. That's what I said. All right. So starting off with the primary growth, but I need a marker. Right. Before starting with the primary growth, let's quickly revise the tissues. What are meristematic tissues? Can you tell me what are meristematic tissues? The online students, I think the chat will be enabled. I think the chat will be enabled. Yeah. Yeah, tell me what are meristematic tissues? Anyone who studied? Anyone who revised? Okay. What are meristomatic tissues? They are undifferentiated tissues which can divide under favorable conditions to produce new cells. They can ask you this definition in your structure essay paper. Okay. What are the three types of meristems we have? Online students, please do reply in the chat. What are the three types of meristems we have? Meristem and intercalary meristem. So out of these three meristems, which meristem is important to the primary growth? Apical meristem. There are two types of apical meristems. What are the two types? Yeah. Shoot apical meristem and root apical meristem. Good. Online students, can I please get a message from you all? Okay. Okay, right. Then what are the characteristics of meristematic tissues? They are living cells. Good. And they have the ability to divide because they are living cells. What else? They are isodiametric in shape. What do we call it? What is isodiametric? It's roughly spherical. Yeah. What about the cytoplasm? No. Thank you. Yeah. So what about the cytoplasm? It has a dense cytoplasm. What about the nucleus? It has a central nucleus. And then talking about the three types of meristems, we have apical meristem, lateral meristem, and intercalary meristems, right? So lateral meristem, what is the main function of lateral meristem? Why are lateral meristems important? Involved in secondary. What are the two types of lateral meristems we have? You can look at the book, no problem, if you have not revised. Vascular can be up and the hope can be up. Good. And what's the importance of intercalary meristems? What's the function of intercalary meristems? Something? Online students, we have Shiva, Jumana. What's the function of intercalary meristems? Rapid regrowth of damaged leaves. Okay? Right. So before we get into the theory section, let me tell you a few, study tips I would say when it comes to biology. Alright. So when it comes to biology, the most important thing is, now I had a friend who had 42 MCQs for biology, still got a C. That means your essay part is as equally important as the MCQs. So just because you have MCQs score like 45 or 46 in your final exam, that doesn't secure you for a good A of the medical faculty. Alright. So when it comes to biology, the main thing is now you can do only the new syllabus past papers, not like physics. Okay, you can't do past papers from 2000 onwards. I mean, that is useless. So you have to do past papers only from 2019 onwards. So you will have around, I think, 8 to 9 papers. No, yeah, around 8 papers. Okay, altogether. Now we had only 5 papers in 2024. So in the new syllabus, you have around 8 papers. So what you have to do is mainly write reviews for each and every MCQ. So how do you write reviews is the next question. Right? Writing reviews, it's not just reading the question. and then writing why is it wrong or why is it correct. Just not one line is not everything. Now, for example, if you take the 2019 paper, can you remember what is the first question of the 2019 paper? The basic question, a very basic question. What is the structural and functional unit of life? That's the question. Okay, this is the first new syllabus year, 2019. So the basic question was, what is the structural and functional unit of life? Where's the answer? Cell. So answer is answer number 3. Now, if I ask you to write a review for this, what would you write? Can I get some suggestions? What would you write? What's your name?
Farade.
Harita. What's your name?
Yes.
So yeah, Lakshmi, what would you write? If I ask you to write a review regarding the cell.
Cells are composed of one or more cells.
Yeah.
Pre-existing cells are formed by existing cells. And what else?
Okay. So when it comes to the cell, when you write about the cell, so you have to say cell is the basic structural and functional unit of life, and you have to explain about the cell theory. Who proposed the cell theory? What are the contents of the cell theory? And when it comes to types of cells, we have 2 types of cells. What are the 2 types of cellular organizations? Yeah, prokaryotic and eukaryotic cellular organization. Then you mention about the prokaryotic and eukaryotic cellular organization. Then you write down the 4 features which are common to both prokaryotic and eukaryotic cellular organization. What are the 4 common features?
Nucleus.
No. Does prokaryotes have nucleus?
I thought it.
No. I think 4 common features.
I didn't comment.
Yeah, common to both prokaryotes and eukaryotes. There are 4 features which are common to these 2 types of cellular organizations. Can I get one from you? Yeah, all the organelles are suspended in the jelly-like material called the cytosol. Whether you consider it a prokaryote or a eukaryote, within the cytoplasm, but you have cytosol. Okay, and all the organelles are suspended in this cytosol, isn't it? You know what a cytosol is, right? Okay, you have a cell like this, and then you have the cell membrane, and inside this jelly-like substance is what you call as a cytosol. What's the other common feature? The outer limit, the cell membrane. Whether it's a prokaryote or a eukaryote, you have this cell membrane. And what else? There are 2 more. Genetic material. What's the genetic material? She's common. Yeah, DNA as the genetic material. In prokaryotes, you have circular DNA. Can you remember? Circular chromosomes. In eukaryotes, we have linear chromosomes, linear DNA. So DNA as a genetic material is common in both prokaryotes and eukaryotes. And what's the final one? Yeah, presence of ribosomes. What is the type of ribosome? 7, yes, ribosome. So these are the 4 features which are common to both prokaryotes and eukaryotes, frequently questioned in your structural essay paper. And also can come under ABCDE MCQs as well. If it comes under ABCDE MCQs, it will be a basic question because that's very easy, because that's at the beginning of unit 2. Okay. But you need to know these 4 features. Now, when you write the answer, for example, if they ask you this question in structural essay paper, and then when you write the answer, for example, if you write the presence of DNA, you won't get marks. In the book, it is presence of DNA as genetic material. So in the structured essay paper, you can't just write one word and expect them to give you full marks. Because the marking scheme is strictly the resource book. So you have to stick to the resource book. Each and every word in the resource book is very, very important. Okay, right. So you have to tell about that. And then I hope you can remember, if you need to, below that section, there is a table comparing the prokaryotes and eukaryotic cellular organization. They have said about the organelles, the diameter, the flagella, the nutritional methods, their chromosome, everything is summarized in a table. Now, how can this come in your question, in your paper? This can come in your paper in a way as an essay. Compare the characteristics between the prokaryotes and eukaryotes. Can you draw a table for an essay? No. You can't tabulate for an essay, you won't get marks. So you need to learn to compare and write. This has this, while this has this. Okay, that's how you write an essay when it comes to a comparison. Okay, so in order to practice that, you practice that whole thing in that one single MCQ. So one single MCQ review will be around 1 CRH. So this is how you write MCQ reviews. Do you have the MCQ? No. Okay. So I'll show you some of my reviews, maybe next week, or I'll send you through the WhatsApp. So I have some of my reviews that I have written for all the 5 years. So take that as an example and don't keep it till the last moment. Now, for example, if you take only Unit 2, from Unit 2, you have around 7 to 8 MCQs in each year. So if you are trying MCQs of 7 years, it will be around 49 to 50 MCQs just from one unit. Now, when it comes to Unit 5, there are around 15 MCQs. Okay, so it will account for around 100 and plus when you consider the whole new syllabus past papers. So if you plan to write all the reviews after you finish the syllabus, that's not going to work. So step by step. Now, for example, this week your work is to take all the Unit 2 MCQs, take separate papers, put the question number, write the reviews. It's okay to keep the resource book while you're writing the review because this is not your exam, right? So it's okay to refer your book and write the review. Do that. That's the main. And also, don't study biology on just one day a week. Your biology is very important. Right. So unfortunately, the thing is, people don't take biology seriously. In our year, the fail rate of, out of the 3 subjects in biology is very high in 2024. Okay, so therefore don't take biology for granted. At least study biology 5 to 6 days a week. That's a must. Okay, I know you all don't do that. Anyone who does that? No. Right? You all study physics, no? Then chemistry. Maybe chemistry because you love chemistry. Is it? Huh? It's not like you don't have to stick to something quite easy, right? You have to practice what's hard for you. Okay? What's your name? Tushari. Tushari. Yeah. Oh, nice. Okay. Right. Without further delay, let's get into the section that we have to start with. Please keep your resource books with you for the cue that's been asked given. Has given a cue for that, all right? So usually, I was talking to your sir, and then I will be providing you all the question here, but not the same note printed again. So please have your resource book with you. Have your brothers and sisters. And please get it out. Any questions that you want to ask? Anything? Am I too fast? Or you can tell me anything. It's fine. Anything you want to tell? Hi. What's your school? Sylvester, you? Sorry? Lexicon. What about you? Ah, both. Okay. So today, first of all, we are going to talk about the primary group. Primary group. So once again, what is the meristem involved in the primary group? Apical meristem, right? You all didn't talk about primary group, right? Are you sure? Which is the last part you completed? So second that we discussed all cells. So did he talk about the primary growth of the root and the shoot? We discussed that. So we don't have to discuss it, right? Has he completed final root of shoot and the root? So then we'll study about the structure, the primary structure of root and the stem. The primary structure of the root and the stem. So talking about the primary structure of the root first. Okay, so we have two types of plants as dicots and monocots. So we are going to study about the structure of these two types of roots regarding the dicot root and the monocot root. Let it be a dicot root or a monocot root. In the cross-sectional leaf, the outermost limit, the outermost structure is going to be the epidermis. The outermost structure is going to be the epidermis. Because we are studying about the structure of the root, root is a structure which absorbs nutrients and water from the soil. For that, there are specialized unicellular structures or unicellular projections that are arising from the epidermis like this. What are these unicellular projections? Which are arising from the epidermis of the root? What are those? These are the structures which absorbs nutrients and water from the soil. What are those structures? Root hair. Root hair. These are the root hairs. So root hairs are one type of specialized cells. These are unicellular projections that is found in the epidermis of a root. So inner to the epidermis, you have the cortex. Cortex is mainly composed of parenchyma cells. Cortex is mainly composed of parenchyma cells. Okay. Now, the innermost layer of the cortex at the center forms a structure called the endodermis. The innermost layer of the cortex forms a single cell layer structure called the endodermis. So usually what is endodermis? Endodermis is the innermost layer of the cortex. Now cortex has multiple layers from the outside to the inwards. The innermost layer is what you call as the endodermis. Okay. Why is it special? Now what happens is sometimes this endodermis, it can deposit a waxy material on its cell layer. A waxy hydrophobic material on its cell layer. What is meant by hydrophobic? What is hydrophobic?
You don't like water.
Yeah, it doesn't like water, or else it is scared of water. Phobia in the sense something, you're scared, right? So it deposits a waxy hydrophobic material on its cell layer and it does not allow water to cross through this. All right. So this endodermis, the cells in the endodermis, they deposit a waxy hydrophobic layer. This waxy hydrophobic layer, we give it a special name. That waxy hydrophobic layer is known as the suberin belt. It is known as the suberin belt because it is composed of an organic compound called suberin. Suberin is a certain organic compound, right? Just like the cellulose or the lignin or hemicellulose, whatever organic compound that you study, just like that. Suberin is also an organic compound. Now, if you have the cell like this, the endodermis, which is surrounding this innermost structure, or as the innermost layer of the cortex, is the endodermis. On the cells of the endodermis, it deposits a waxy hydrophobic layer called suberin. Okay, so this structure, this waxy hydrophobic layer, we call it by a special name. We give it a special name called the Casparian strip. Casparian strip. Okay, so what happens is, once again, the outermost layer is the endodermis, and inner to the endodermis you have the cortex, and the innermost layer of the cortex is the endodermis. The epidermis, cortex, and the innermost layer of the cortex is the endodermis. The cells in the endodermis, they deposit a waxy hydrophobic layer formed from the organic compound called suberin, and that belt is known as the Casparian strip. Okay, now actually what happens is, now this is a root, right? This is a root. Now, water is absorbed through the root hairs, and the absorbed water travels through this cortex and reaches this point called the endodermis. All right, you understand it, right? The root hairs absorb water and minerals from soil solution, and then it is transported through these cortex until this point. Now, can it cross this point and go inside? No, because on the cell layer of the endodermis, it has deposited this suberin belt. So therefore, it has to follow another pathway to go inside. Okay, this pathway where water is transported from the root hairs up to this point is a specialized pathway called the apoplastic pathway, which we'll be discussing later. Apoplastic pathway. For now, you need not to know what an apoplastic pathway is. Okay, so what you should know is when the water is transported up to the inner layer of the endodermis, from that point onwards, it is blocked. All right, so therefore, it has to follow another pathway in order to enter the inside. Because inside we have the vascular cylinder. What are the vascular tissues we have? Xylem and phloem. Water is transported towards the upwards, the shoots and the leaves through this xylem. So the water that is absorbed from the root hairs somehow has to reach the xylem. Okay, now this is not the xylem, there are many structures inside, but xylem is also inside. All right, so somehow it has to reach the xylem. But if it is blocked from here, it has to follow another pathway to reach that xylem. Okay. Now, inner to this endodermis, there is another structure which is composed of at least 2 or 3 cell layers. Okay, inner to the endodermis, there is another structure which is composed of at least 2 or 3 cell layers, which is called the pericycle. Which is called the pericycle. Okay. We'll talk about the pericycle later, but now remember, it is composed of parenchyma cells and it is at least 2 or 3 cell layers. Okay. Inner to the pericycle, we have the vascular tissues that we were discussing a while ago. What are the vascular tissues? Xylem and phloem. Okay. So, in dicot roots, in dicots, the xylem is located like a star inside this area. It looks like a star. I know this doesn't look like a star, but it looks like a star, the real structure. Okay, this is the area which comprises the xylem tissue. Then where is the phloem? Phloem is in these grooves of the arms of xylem. Now these are the arms of xylem. The arms of xylem forms a groove with the pericycle. Can you see that? The arms of xylem, it forms a groove with the pericycle. So these are the places where you can find the phloem tissue. Okay, what's the function of phloem tissue? Transportation of food, basically, simple as that. Okay, transportation of sucrose, technically, but we can say transportation of food. So once again, I'll tell you now. Forget about the monocot root because the monocot root structure inside is a bit different. Okay, so we'll be studying about the monocot root later, but get the basic idea first of all. So first of all, what do you have? You have the epidermis, like this. Epidermis is the outermost covering. Okay, and on the epidermis, there are specialized unicellular projections called what? The root hairs. What's the function of root hairs? Absorption of water and minerals from the soil solution. And inner to the epidermis, this area is the cortex. The innermost layer of the cortex, is what you call as the endodermis. Okay, the innermost layer of the cortex is what you call as the endodermis. And with this endodermis, it deposits a waxy hydrophobic material called suberin on its cell wall. Okay, Now, suberin is waxy and hydrophobic. The belt that is formed from the suberin is known by a special name called the Casparian strip. Is that clear? Ashu? You can ask any doubts, you can let me know. Anything is welcome, criticisms also. So you have a Casparian strip. Is that clear? When it comes to the endodermis, the endodermal cell walls, it deposits a waxy material called suberin, which is hydrophobic. That belt that is formed from the suberin on the walls of the endodermis is what you call as the Casparian strip. Sure. Sure. Okay. If you have any doubts, let me know. Then inner to the endodermis, we have the pericycle. So pericycle is formed at least from 2 or 3 layers. At least from 2 or 3 layers. So let me draw 2 layers of pericycle. Okay. So because we're talking about the dicot roots, remember in dicot roots, this pericycle has a meristematic function. Meristematic function. Now, this pericycle, which is found inner to the endodermis, is at least composed of 2 or 3 cell layers, and it is mostly made out of parenchyma. Parenchyma. Mainly, it is made out of parenchyma, and its dicots only has a meristematic function. Meristematic function in the sense it has the ability to divide only in dicots. Yeah, only in dicots it has the ability to divide. Because we study about the secondary growth next. In secondary growth, can you remember when you studied about the lateral meristems, you studied about 2 types of cambiums, the vascular cambium and the cork cambium. Okay, these dicots, they undergo secondary growth. So when they undergo secondary growth, their cork cambium in the root is formed from the pericycle. Their cork cambium in the root is formed from the pericycle, which we will be studying next, later. Okay. So for now, remember the pericycle in the dicots, they have a meristematic function, which means it has the ability to divide, and it involves in secondary growth. Because monocots, they do not undergo secondary growth. Only dicots, they do undergo secondary growth. Okay, right. Then inner to the pericycle, we have the vascular cylinder. Vascular cylinder consists of what and what? The xylem and the phloem. In dicots, the vascular cylinder forms a star-shaped structure like this with grooves. A star-shaped structure like this with grooves. So this area entirely is the vascular cylinder. Okay, means xylem plus phloem. All right. So where is exactly the xylem and where is exactly the phloem? Now this area, which looks like a star, a solid core in the center exactly, is the xylem. Okay. And with the arms of this xylem, it forms grooves like this. Grooves can be hilascated. See, I don't know the Tamil word. Okay. Grooves can be hilascated. So these grooves that are formed with the arms of this xylem, they are the places where you find the primary phloem tissue. Okay. Is that clear? Sure. And I said your dicots undergo secondary growth, isn't it? Right? You understand that, right? Dicots undergo secondary growth. Okay. Not all plants undergo secondary growth. Okay. Many dicots, not all dicots either. Many dicots undergo secondary growth. And so now if they undergo secondary growth, you have studied lateral meristems. There are 2 types, vascular cambium and the cork cambium, isn't it? So I said you, in the roots, the cork cambium is formed by the pericycle. So where's the vascular cambium? The vascular cambium is located between the phloem and the xylem like this. This is where you find the vascular cambium in the dicot of the root. root of the diaphragm. Okay, so here is where you find the vascular cambium. So always remember, vascular cambium is located in between the xylem and the phloem. Xylem, phloem, after the vascular cambium is the beginning. Okay, it's like this. If I draw this structure separately here, you have star-shaped structure, and then you have the cambium like this. Okay, you have the cambium like this, and then this area which forms with the pericycle is the phloem. Okay, remember cambium, this vascular cambium that is found in between the xylem and the phloem is found only in dicot roots, not in monocot roots. Monocots do not have cambium. They do not produce lateral meristems because they do not undergo secondary growth. Is that clear? Are you sure? Any doubts? Any doubts? Doubts there. Doubts there. What is the function of let me ask like this? What is the function of root hairs from lumping?
Absorb the nutrients from the soil.
Absorb water, designating water and nutrients from the soil solution. And then what is the organic compound that is deposited on the endodermal cell walls? Honey. Sugar. Sugar is the organic compound. And what's your name?
Ali.
Sorry?
Ali.
Ali. Okay. So I'll ask you this question. Yeah. Pericycle is mainly composed of which types of cells? Pericycle is mainly composed of which types of cells? No. Xylem and phloem is the vascular cylinder inside. The outside structure is the pericycle. I'm asking about the pericycle. What's your answer? Okay. Cortex is also mainly made out of parenchyma. The pericycle is also mainly composed of parenchyma. Okay, parenchyma cells. All right. Yes. Do you want me to explain this again? All right. We'll read through the resource book and then I'll quickly explain it once again. All right. Okay. So, do you have the resource books? No. In that, this part is not there. Oh, look at this part is there. So, has the given this to everyone? Oh, there is an activity there. We can do that as well. So once again, I'll quickly explain you. Okay, before I explain, now tell me whether you have any issues. Like you can tell me anything. Okay, I'm just 2 or 3 years here to you, so yeah. Anything that you want me to improve, because like this is class I was asked to take up. So because of that reason, I know you all are facing a difficulty, like when the teachers, which I understand that you all are facing a difficulty. And I'm just a first year medical student and just I entered the medical faculty. So I might not have better teaching methods like yours do, but I think I'm trying my best. So yeah, any feedbacks that you want to tell me? Anything? Criticisms are not from you. No problem at all. You can be very friendly and talk to me. Online students, can you please reply me in the chat? Any doubts you have? Or anything that you want to tell? Is that clear? Assured? Anything that you want to say? Anything you want me to improve in teaching the next section? Nothing? Right. So once again, I'll quickly explain and then we'll read through the resource book and then we'll finish off the diagram part. All right. So once again, We're talking about the primary structure of the dicot roots and the monocot roots. So first of all, we are going to talk about the primary structure of the dicot roots. Okay, so the outermost limit of the dicot root or the monocot root is what? The epidermis. Okay, in the epidermis there are the hair-like projections called the unicellular projections, which are known as root hairs, which absorbs nutrients and water from the soil solutions. Remember, these are unicellular projections. Okay, on the epidermis, you have hemicellular projections called the root hairs. Root hairs are not present on the shoot. Okay, it's present only in the root. Now, because you have completed epidermis with your last class, tell me what are the other two special structures found in the epidermis? In the epidermis, there are three types of special structures. Okay, one is this root hairs. There are other two structures. What are the two other structures? Special structures? Hmm? No. I'm asking. On the epidermis, you have studied the structure of the epidermis. You have studied the functions of the epidermis, right? During your last class, you said that he has already completed those sections to you. Right? So, in the epidermis, there are three types of special structures. One is these root hairs. There are two others. Huh? Guard cells, good. One is guard cell, what is other? Trichomes. You can remember. Good. Guard cells, trichomes, and root hairs. Have you heard about them? Mother. Mother. It is the analysis book. Have you revised? I am Bible again. Yeah, take away Bible and nothing of that. Trichomes क्या होना? Harit, Matagaran. Ali, what are trichomes? What are guard cells? Basic knowledge, what are guard cells? Can you all remember those three structures? I can't remember the name. Sanjay, can you remember those three structures? Okay, what are the functions of trichomes then? Yeah, good. Mosaic has replied root hairs, trichomes, and guard cells. Online students, you all can reply. Tell me what are the functions of trichomes? There are two types of trichomes. Do you know that at least? Chaotic trichomes, the trichomes which secrete chemicals. How they know? Can you remember? No. Huh? What are the functions of hair-like trichomes? There are two functions. Colors can come. So, what are root hairs? Not hair-like trichomes.
Shiny hair reflects excess sunlight.
Yeah, good. One is shiny hair can reflect excess sunlight. What is the other function? What is the other function? What like students? Shima, the same good. Reduce water loss. So remember, no, epidermis in the now when it comes to epidermis, in the epidermis there are three types of special structures. One is the guard cells, the other one is these root hairs that we discussed, and the other one is the trichomes. What are the root hairs? They are unicellular projection. Unicellular can have the ecosellaki. Can you understand Singhal? If I say like that, yeah, right. Unicellular projections, and what is the function? They absorb water and minerals from the soil solution. Then we have guard cells. Guard cells can go. Guard cells can go. Have you heard of the structure of the stomata? Stomata, what is the number? Stomata can what is structure is stomata a structure or is it a hole? Can you palpate the stomata? Can you define a structure for the stomata? No, right? Sorry? Actually, it's a hole, okay? What opens and closes the guard cells. Alright? Now, when it comes to stomata, when we study as stomata can get a hole not bigger. There is a dough-like structure which is surrounding the stomata. For example, suppose this is the stomata you talk about, this is the core. Surrounding the stomata, you have structures called the guard cells like this. The guard cells, the way they open and close, just like a door. When they close, stomata is closed because stomata is this core. And when they open, stomata is open. What is the function of guard cells? Gaseous exchange. Because when it is closed, it blocks gaseous exchange. When it is open, it allows gaseous exchange. Is that clear? And there is a specialty about guard cells. When you compare with other structures in the epidermis, there's something special. There's something unique to guard cells. They can ask that. What's a unique feature of the guard cells comparing to the other epidermal cells? What is a unique feature of the guard cells? Online students? Something, what is it? You watched my recording, right? Huh? Didn't you guys? Huh? What's the unique feature? Something check the book and say. It's there in your resource book. It's the only type of epidermal cells which contains chloroplast. The guard cells are the chloroplast came out. Why chloroplasts are required for photosynthesis? You have learned photosynthesis, right? Okay, you have learned photosynthesis. Photosynthesis produces ATP energy through light reactions. In order to open and close guard cells, in order to do that process, there is a separate process. We'll be learning about that later. Do that process, we require ATP energy. That energy is provided by the light reactions that are happening inside the chloroplast of these guard cells. So guard cells are the only epidermal cells which contains chloroplast, which means they are the only epidermal cells which can carry out photosynthesis. Is that clear? Right? Then we have two types of trichomes, hair-like trichomes. There are two functions. One is reducing border loss. The other function is, as she said, shiny hairs reflect excess light. Okay? And then we have the trichomes which secrete chemicals. These chemicals provide defense against various insects, herbivores, likewise. Okay, so these are the three types of special cells in the epidermis: their structure and their function. They can ask you to write a short note on epidermis. If you don't write all this, you will get marks. No. They can ask this instruction is very probable and very easy to score. You should be able to answer very quickly, like instantly, because these are the basics. Have you even check the grade 13 book? No, right? Grade 13 resource book. Starting from unit 7 onwards, all you have to do is memorize without the logic. Memorizing without the logic. These are memorizing with the logic. Trust me, memorizing without logic is 100 times harder than memorizing with the logic. So if you don't do this now, you won't be able to go for a good result. Because these are the basics. Now, in my A-levels journey, I did A-levels in 1 year and 2 months. So, in my A-levels journey, I have gone through this first resource book at least 12 times. Minimum of 12 times, right? Fully start again and again and again. Okay, so that's why I can remember. That doesn't mean like I have revised in the morning. I had a separate session in the morning. So, yeah. You have to revise. Okay. When you just if you revise once after your class, that's not enough. In order to retain that in your long-term memory, you have to repeat it like at least with 3-day intervals, 5-day intervals, and then in a week, and then in a month. Okay, you have to do that. All right. Okay. I was distracted. I was teaching about the structure, right? So once again, regarding the Dicot root, what is the outermost structure? Epidermis. In the epidermis, we have root hairs, which are unicellular projections, right? By the way. All right. Inner to the epidermis, we have the cortex, right? The cortex is mainly composed of parenchyma. Okay. The innermost layer of the cortex is what? Innermost layer of cortex is what? Endodermis. The innermost layer of the cortex is the endodermis. The endodermis is a single cell layer, okay, because it is the innermost layer. All right, and it deposits a waxy hydrophobic material called suberin. The belt that is formed out of suberin is called a Casparian strip. All right, so it blocks the water transport from here to here and then entering from this point to inside. All right, so we call this process as apoplast. Actually, this for now only this word is important, but you don't need to know the procedure because we'll be learning about the radial transportation. Okay, so just remember this part where water is transported through the cortex. It is called cortical apoplast. Cortical apoplast. That is the process where the root hairs absorb water and minerals, and those water and minerals are transported up to the endodermis. Till that point, that process is called the cortical apoplast. And from that point onwards, it can't cross the endodermis. Why? Because the endodermis has deposited a waxy hydrophobic material. So because it can't cross the endodermis, through the apoplastic pathway, apoplast is one type of water transporting pathway. Okay, because it can't cross the endodermis through the apoplastic pathway, it follows some other pathway called the symplast, which you don't have to know, and enters the vascular cylinder. And then again, inside the vascular cylinder, water is transported by a apoplastic pathway again. We call that apoplast as vascular apoplast. Vascular apoplast. So what is the function of endodermis? Endodermis blocks the cortical apoplast from the vascular apoplast because it's like a barrier between vascular apoplast and cortical apoplast. Apoplast I can or even because you don't have to know it's a complex position and we're learning later. But now just remember that apoplast it has a lot of things. What are the contents in it and how this water transported. So for now, I'm not going to explain you what is apoplast. So for now, remember, endodermis blocks the water transportation at this point. So because we have to explain it in a proper way, it blocks the cortical apoplast from the vascular apoplast. Remember that. Okay. Right. In order to the endodermis, we have the pericycle. Pericycle is composed of parenchyma cells. Pericycle is at least composed of 2 layers or more, 2 or 3 layers. Okay, endodermis can a single layer. Pericycle can at least they have khotuna. Why I am emphasizing this again and again is make a daragena when they say pericycle is a single layer of cells composed of parenchyma, pericycle is at least composed of 2 or 3 cell layers. Okay. And then remember in Dicots, the pericycle has the meristematic function, which means it has the ability to divide and it involves its secondary group. Inner to the pericycle, we have the vascular cylinder. Vascular cylinder is composed of xylem and phloem mainly. Because dicots undergo secondary growth, there is a vascular cambium as well in the vascular cylinder. So the star-shaped area is the xylem, and in the grooves you have the phloem, and then in between the xylem and phloem we have the vascular cambium. Okay. Clear? Right. Now, having this idea, we'll study about the monocot root structure cell. Alright, it's nothing much. The change happens after the section of endodermis. Okay, so studying about the monocot root, in the monocot root also, the outermost structure is the epidermis. Okay, epidermis has root hairs, epidermis has root hairs, which absorbs water and minerals from the soil solution. Okay, inner to the epidermis, we have the cortex. Okay, and then the cortex continues until the innermost layer of the cortex called the endodermis. And endodermis, similar to here, it has a Casparian strip there also. Okay, inner to that, we have pericycle once again. So here pericycle is also composed of 2 or 3 cell layers made out of parenchyma. Alright, and the speciality is this pericycle has the meristematic function. This pericycle does not have the meristematic function. They can monocot root take a pericycle and divide when the fat. Is that clear? Because monocots do not undergo secondary growth. Alright. Many diamonds undergo secondary growth, but monocots do not undergo secondary growth. So this pericycle does not have the meristematic function. Okay. Then, inner to the pericycle only, you can find the vascular cylinder just like here, but the structure is different. Here, you have alternating ring of vascular cylinder like this, alternating ring of xylem and phloem like this. So this is how xylem and phloem is arranged. It is an alternating ring. A ring is arranged inside the vascular cylinder. The xylem and phloem is arranged in a ring. Xylem, phloem, as an alternating ring. There is no proper definite structure as this. So when you are describing about the monocot vascular cylinder in the root, you have to say it is an alternating ring of xylem and phloem. Alternating here again, xylem, phloem. One after the other, and it is repeating. Okay, it is an alternating ring of xylem and phloem like this. And here there is xylem and phloem with the katha vivarai. But here there is a area. Here there is a remaining area. That area is also a part of cortex, which is composed of parenchyma cells. We don't call it the cortex, we call it the core of parenchyma cells. It is a central core of parenchyma cells. Okay, again, exactly at the center, right? A central core of parenchyma cells is situated in the vascular cylinder. Okay, and another thing is, it does not have vascular cambium. Here, in between xylem and phloem, there is vascular cambium. In monocot roots, there's no vascular cambium because there's no use of having vascular cambium because it does not undergo secondary growth. Is that clear? Sure. Right. Now, can I go through the context? You don't have to book, right? Oh. Any doubts? Online students, any doubts? Is the board clear as well? Read through your resource book, page number 116. You know, you can refer this to some videos download. When will you start paper class, you know, I'm not sure I'll let you know. Um, I've not even thought about that because today's my first day. I'll let you know soon. Yeah. Yeah. Please study cutting hair off, Mama. You all have to remember. None of you can remember anything you studied in the last class. Except a few things. Then the answer I not could study. Because because I did it was in 1 year and 2 months. Sometimes I had to study for 15, 17 hours a day as well. 8 hours is not it. Done? Any doubts? Any doubts? No. Any file line number? No. Usually the cell gives the break up to 2 hours. Is it like this? Yeah. So at what time do you want to break at 10? Farade, and what time do you want to break at 10? Walk. Per day, how many hours should we do self studies? You know, 7, 8 hours? We have classes 6 hours is enough. Shall we start the next section? Do you have any doubts? Ali?
So we are in the same hall, especially for.
It's for the root, because it's the primary structure of the root. Next, we are going to study about the primary structure of the stem. Okay? Because root has that. Sometimes they can give you this structure in your the diagram in your structure display paper and ask you to recognize this diagram and also to label it. Okay, so when you are recognizing and when you are writing down what is this what this diagram is, you can't just write this is the Dicot root or the Monocot root. You have to say it is a longitudinal section of the Dicot root or the cross sectional view of the Monocot root likewise. Because Monocot root, when you look from the morphology, when you look from outside, does it look like this? All right.
That's pure and it makes the growth of that a cycle.
No. This
So what makes the growth?
which growth?
I mean, like you said, the monocots, monocot dicots, the roots are both are similar.
Yeah.
So why don't they grow?
You mean the secondary group?
No, you said how can I say that part? You said that part doesn't grow in monocots.
Yeah, vascular Kandy is not present in monocots.
So is it due to vascular Kandy?
No, Now, vascular cambium is one structure. Okay? Now, there is two types of growth, primary growth and secondary growth. Certain plants undergo only primary growth. Certain plants, in order to increase their diameter, they undergo secondary growth. That's what's happening. So, not all the plants undergo secondary growth. Those which undergo secondary growth has this vascular cambium and the fork cambium. Okay. So, when it comes to monocots, they do not undergo secondary growth. They do not increase their diameter. They are ecoseric hadronama, that's it. Okay, they do not undergo secondary growth and increase their diameter. Many of them, they don't grow. They grow only, vertically, not horizontal. Secondary growth is important. The plant grows horizontal, basically increasing the diameter. If you compare between a hopkins tree and a mango tree, you can obviously see two differences. The girth of a mango tree is really high because it undergoes secondary growth. But when it comes to a coconut, from top to bottom, it almost has the same diameter because it does not undergo secondary growth. Okay. So vascular cambium is a structure that is important for secondary growth, not for the primary growth. So for the plants which do not undergo secondary growth, there is no use of having this. That does not affect the main growth because this is an additional growth that happens in certain plants. Is that clear? Right. So next we have to study about the primary structure of the dicot plant stem and the monocot plant stem. Okay. So when it comes to the plant stem also, the outermost layer is epidermis. This epidermis is important to prevent desiccation as well as to prevent infections because now roots are present under the ground level, below the ground level. But these aerial parts, the stem is an aerial part, which means it is exposed to the outside. It is above the ground. So it is exposed to the sunlight, which means there's high probability that due to the sunlight, it can undergo desiccation. What is desiccation? Dehydration. It can undergo dehydration. So, in order to prevent the plant from undergoing dehydration, this epidermis plays an important role. And also, just like us, in us, infections are mostly prevented by your skin, the barrier, because skin acts as a physical barrier. Just like that, epidermis is a structure which helps to prevent infections in plants. It does not 100% prevent, but it provides a maximum protection to prevent infections in plants. Okay, so epidermis is important in aerial parts, basically the stem, because it prevents desiccation as well as it prevents infections. Okay, and because this is an aerial structure, because aerial in the sense it's above the ground, these epidermis, it contains structures called the stomata. It contains structures called stomata. Remember, gaseous exchange does not happen only through the leaves. Gaseous exchange happens only through the leaves here. Leaves are even in the epidermis you have stomata. So the stomata is important for gaseous exchange. Okay. Is it clear? Sure. And does this have root hairs? The epidermis? No. Any doubts? Roots have root hairs. Root hairs are not important for the stem. So remember, in the stem, the outermost layer is the epidermis, which prevents from desiccation and infection. And this epidermis has pores called stomata, which is important for the gaseous exchange. Okay. Inner to the epidermis, this entire layer is the ground tissue layer. Okay. I think you have studied about the ground tissue, epidermis, the vascular tissue, right? You all studied about the structures now, because usually in a plant, you have the vascular tissue layer at the center. Surrounding that, you have the ground tissue layer, and the outermost layer is the epidermis. That is the basic structure. Okay, so now this is the epidermis. Inner to the epidermis, what do you have? You have the ground tissue layer. Okay, In dicots, this ground tissue, now we are talking about the structure of the dicot stem, okay? In dicots, this ground tissue is differentiated into cortex and pith. Cortex and pith. Ground tissue is mainly composed of parenchyma cells, mainly. Okay, majority of its content is parenchyma cells, and this is differentiated into two different structures for the cortex and pith. So inner to the epidermis, this area what you find here is the cortex. Okay, just inner to the epidermis, not this entire area, but this the outermost circle. There's no special like boundary in between. So this outermost part is what you call as the cortex. Okay, and this central area is what you call as the pith. This entire thing is the ground tissue. The ground tissue is differentiated into two things as the cortex and the pith. Okay, now this area is the cortex, and at the center you have the pith. That happens only in dicot roots. Okay, that's why dicot stems. Alright, so once again, you have the epidermis. Epidermis is interrupted by stomata. Inner to the epidermis, you have the ground tissue layer. The ground tissue layer is differentiated into the cortex and the pith. Okay. Okay. Alright. Additionally, you also have studied about collenchyma, right? Have you? But collenchyma? What's the function of collenchyma? What's the function of collenchyma? What's the function? It has only one function. What is the function of strength support. It's not accurate enough, but it does not involve in photosynthesis. That's all. It provides mechanical support, that's correct, but it should be more accurate. Is collenchyma a living or a dead tissue? Is it living or dead? Dead. There was a function. It provides mechanical support without restraining the growth, which means now if you consider sclerenchyma also provides support. Okay, but the thing is, sclerenchyma is a dead structure. Okay, so suppose if this is a certain plant part, and here you have a layer of sclerenchyma to provide support, this part cannot grow further. Okay, because while providing support, it does not allow this part to grow further. It limits the growth. But when it comes to collenchyma, suppose this is collenchyma, it will provide mechanical support along with the growth of the plant, it also grows. So without restraining the growth, without blocking the growth, it provides support. Collenchyma provides support without blocking the growth. But sclerenchyma, when it provides support, it does not promote growth. Is that clear? Okay. Would you have any doubts? Sure. Okay, so now apart from parenchyma, we also have collenchyma and sclerenchyma here. Okay, just beneath the epidermis, just below to the epidermis, you have strands of collenchyma like this to provide additional support. Have strands of collenchyma to provide additional support. Apart from that, in the cortex, you also can find sclerenchyma, which the location is not specified. Okay, sclerenchyma is also providing the cortex to provide additional support. So the ground tissue is mainly composed of parenchyma. It is differentiated in the cortex and the pith. Here the outermost part is the cortex, and in the middle we have the pith. Just beneath the epidermis, We have what? We have parenchyma strands to provide support. Also in the cortex, additionally, we have sclerenchyma to provide additional support as well. Okay, so the majority is parenchyma. Apart from that, in the cortex, you have collenchyma as well as sclerenchyma. So when you're explaining about the collenchyma, you have to properly say that there are strands of collenchyma just beneath the epidermis. There are strands, again, strands, I don't know, signal about. There are strands of collenchyma just beneath the epidermis, just below to the epidermis. These locations, these words are very important in order to provide support. Okay, so now we have discussed about the cortex, the pith, as well as the epidermis. Now we have to talk about the vascular cylinder. So when we talk about the vascular cylinder in Dicot cell, it consists of a ring. It is a properly arranged ring. Okay, so the ring looks like this. It has the xylem, the phloem, as well as the vascular cambium. Why does it have the vascular cambium? Because it undergoes secondary growth. So always remember, vascular cambium is located between the xylem and the phloem. Now, when we talk about this dicot stem, in the dicot stem, the xylem is facing the pith. Now, this area is the pith now. So, xylem is facing the pith and the phloem is facing the cortex. In between the xylem and the phloem, you have the vascular cambium like this. Okay. Now, this is your phloem and this is your xylem. In between the two, you have the vascular cambium. Okay, so this structure is arranged as a ring all over. This structure is arranged as a ring all over like this. It's a proper symmetrical ring. Like this. It contains the xylem, phloem, and the vascular cambium. Always remember, xylem is facing the pith and cortex, or the phloem is facing the cortex. Phloem is the outer structure and xylem is the inner structure. So this is in the dicot stem. So once again, I'll tell you, in the dicot stem, we have the epidermis. Epidermis is affected by stomata, and then inner to the stomata, inner to the epidermis, we have the ground tissue layer. Ground tissue layer is mainly composed of parenchyma cells. It is differentiated into cortex and pith. Here we have the cortex at the center of the vascular cylinder. This is the vascular cylinder. At the center we have the pith. Okay. In the cortex, there are strands of parenchyma to provide additional support. Apart from that, there are sclerenchyma as well. Okay. I'm talking about the vascular cylinder. In the vascular cylinder, you have xylem and phloem. Xylem is facing the pith and phloem is facing the cortex. In between the xylem and the phloem, we have the vascular cambium. Okay, and surrounding it, now this entire thing is what you call as a vascular bundle. This entire thing is what you call as a vascular bundle. Surrounding the bundle of surrounding the vascular bundle, basically xylem and phloem, there are sclerenchyma cells to provide support to the vascular bundles. Surrounding the vascular bundles like this, there are sclerenchyma cells, clusters of sclerenchyma cells, a lot of sclerenchyma cells like this to provide support to the vascular bundle. Is that clear? Is that clear? Sure. Right. Can you go through the resource book section again? Yeah. Right. This is more statement there saying lateral shoots develop from axillary buds. Okay. Windows. Okay. So when it comes to plant, in a plant like this, we have these, right? These areas are called axilla. The plants. These areas are called axilla. In these areas, there are small buds called the axillary buds. These axillary buds can give off to branches. So lateral shoots are basically branches. Lateral in the sense in this direction. Okay, so these axillary buds they give rise to branches, which we scientifically say as lateral shoots. Okay, so in dicot stem, in the dicot plant, in the dicot stem, we have axillary buds. The axilla is the area at the petiole and the stem, this area. Okay, so these areas have buds called the axillary buds, and these axillary buds they give off to lateral branches. Okay. Clear? They have not questioned about that till date. So yeah, it's just a small point. Any other doubt regarding the dicot stem? No? Yeah. You want to know what is the question? No, it's specific. It's now so this is the cluster of sclerodermosis. Okay, surrounding the vascular. Yes, cluster of sclerodermosis. Right. Regarding the monocot stem, it's nothing much. Okay, in the monocot stem, we have the epidermis like this, and inner to the epidermis, what do you have? Now, epidermis anyways have stomata. Okay, let it be a monocot or a dicot, it contains stomata. Inner to the epidermis, we have the ground tissue layer. Remember, in monocots, the ground tissue layer is not differentiated into cortex and pith. So, this is just the ground tissue layer. Okay, this is not differentiated into cortex and pith in monocots. Differentiated in the sense it is like developed in their structure and function. Okay, it is more evolved compared to the monocots. That's why we call as differentiated. So here it is advanced, more advanced compared to this. The ground tissue is more advanced here because it is differentiated into cortex and pith. Okay, here the ground tissue is not advanced, it is primitive compared to this. The ground tissue is not differentiated into two different structures. It is in its primary level. Okay, so here this is composed of the ground tissue. The ground tissue is composed of parenchyma cells, and here you have vascular bundles which contains the xylem and the phloem, which is surrounded by the cluster of sclerenchyma cells. But there is no proper arrangement as such in here. Here the vascular bundles are arranged in a ring. Here, the vascular bundles are scattered throughout the ground tissue. You have the vascular bundles everywhere scattered without any proper arrangement. So, a vascular bundle, one vascular bundle, it consists of the xylem, the phloem, and the cluster of sclerenchyma cells. Does it have the vascular cambium? No. Because they are monocots, they do not undergo secondary growth, so they do not require this vascular cambium. So the vascular bundles, it has the xylem, phloem, and the cluster of sclerenchyma cells that is surrounding it, and they are established throughout the ground tissue. That's it regarding the Monocot step. Is that clear? Ground tissue is not differentiated into cortex and pit. The vascular bundles are scattered throughout the ground tissue. The vascular bundle does not contain a vascular cambium. At the same time, each vascular bundle, just like in there, it is surrounded by a cluster of sclerotic cells to provide support. There. There. Dots. And go through that section. Look through the diagrams very clearly because I'm going to ask one of you to come to the board. Would you mind if I finish the class a little early because yesterday I went through a small accident and I have my leg bandaged. It's really hard for me to stand on stick. Please look if I finish by now. Yes. Sure. Do you have past papers? Do you bring past papers to class? No? Oh. Can I write down these questions? I'll be suddenly giving questions from maybe unit 2, unit 3, like a quiz also. No, this is from unit 4. You can refer the book for this, no problem. So prepare for unit 2, unit 3 questions. Right. First question, what is known as a tissue? Just roughly write it down very quickly. What is known as a tissue? Number 2, what are meristematic tissues? Number 3, what are the characteristics of meristematic tissues? Next one. What are the 3 overlapping zones of meristems? What is known as the primary growth of the root? Next one. What happens to the cells? What happens to the cells? Produced outwards to the root apical meristem. What happens to the cells? Produced outwards to the root apical meristem. And what is the importance of that structure? And what is the importance of that structure? Next one. Mention two differences between the shoot apex and the root apex. Next one. Mention two features in. Mention two features in. Collenchyma tissue. Collenchyma tissue. That differs from parenchyma tissue. That differs from parenchyma tissue. What are the two types of sclerenchyma cells? And comment about their structure. Next one. What's the importance? What is the importance of having? What is the importance of having? Secondary cell walls. Secondary cell walls. Thickened by lignin. Thickened by lignin. You have studied all this stuff. Thickened by lignin. In vessel elements and tracheates. In vessel elements and tracheates. Next one. What are sieve cells? What are sieve cells? Which type of plants? Same question. Which type of plants consist of sieve cells? What is the structure which connects? Next question. What is the structure which connects the sieve tube element and companion cell? Which structure blocks the cortical apoplast from the vascular apoplast? Apoplast to right apoplast. Which structure blocks the cortical apoplast from the vascular apoplast? Next one. What are the two main functions of the root cortex? What are the two main functions of the root cortex? Drouin-Level, the primary structures of dicot plant stem and monocot plant stem. Final question. Mention two differences between dicot plant stem and monocot plant stem. I think there are around 16 questions, right? I'll give you around 20 minutes. You can refer the resource book. But when you're drawing the structures and stuff that we learned today, I mean, the things that we learned today, try to write from your memory rather than referring to the resource book. Maybe the earlier ones, because you haven't revised or you can't remember. Well, if you need to, you can do this. Okay. Process. You are having the latest resource book, right? Sure. Some of Thank you. Now we use the old one and it can be so much information. Right. It is fine. So when I have asked the definition and all, I'm expecting you to write as if you are writing a structure listing, which means you have to stick to the resource. So for example, in the resource book, if it says group of one or more cells, and if you write a collection of one or more cells, mostly you don't get marks, even though it is good. Unfortunately, that's the problem. Please drop your message. Got it. Any doubts that you all came across? So sorry for the interruption in the middle. And an additional thing I wanted to show you all. So I don't know how far it's effective for you all, but for me, I used a lot of charts and diagrams to, memorize stuff which are harder to memorize. Now, for example, you all have studied unit 3, right? So you have studied all the five, starting from Hidaya, right? So for that, I used to prepare charts like this. I don't know the audience will see this. Yeah, they can see. Anyways, so these are like charts that I prepared, like demonstrating the structures. And when it comes to platyhelminths, winters, they have their own body structure and they have eye spots. Likewise, drawing everything and taking it into diagram. So you have final and everything. So this is better to be done, especially for the last few units rather than the first units, because the first few units are easy to memorize, but the last units are not that easy to memorize. So I'll just pass this book through you all. You can go through it. And yeah, if you want, you can do this as well. Okay. Online students have you completed Fatima, Rima, Aksha, Natasha. Aksha, Natasha. And we have, who else do we have? Hussein and Sudiksha, have yours completed? And introduce yourself to me, like where are you from? Without a break, I'll continue to the class again till 11. And have a long break after 11. Everyone of you here wants to enter medical faculty. I mean, different ideas. Something won't get medical faculty. But what, like everyone wants to become doctor? No? So what's your plan? They are very shy though. Like, in our classes, for example, in Nilem's class, he was fed up of us talking to him. Because we always disturb him talking without letting him take marks. Yeah. Yeah, don't even talk. Those who go to electricity physics classes. Online students, have you completed? Like, they have done. Can I drop you a message? Have you all completed? I'll give it 5 more minutes and then we'll start discussing. Can anyone of you give me a WhatsApp number so that I can send? I have a unique table where it's a compilation of all the along school papers MCQs. So I can forward it to you and we can discuss it meanwhile if you get time. Because those are very good MCQs, kind of hard ones that gives a very good practice. Can I get any one of your? One more second. 3 more minutes. How did you guys do the or- I think that's it. Right. Okay, whatever. Right. Completed. How many questions more? Maybe 3 more minutes to complete. Maybe almost 20 minutes. The site have been completed, so please Shalavi and Natasha and this one more partner have well completed. Okay, we'll discuss. Are we? Okay, what's the first question? What are tissues? What are tissues? It is a group of one or more cell types which is specialized to perform a certain function. It is a group of one or more cell types which is specialized to perform a specific or a certain function. Is what you call as a tissue. Have you written that? It's almost similar idea. We'll also check the exact wording in the resource book. It is a group of one or more cell types which carries out a specialized function. We have written that exactly, then that's correct. Next one. What's the next question? What's the next question? What are meristematic tissues? Lucky to tell the answer. You did right. Hurry.
What's features?
Call meristems. Is that all you wrote?
Which consist of constantly divided and suitable functions and
Okay, so meristematic tissues, they are undifferentiated tissues found in the plants which can divide constantly under suitable conditions to produce new cells. They are meristematic tissues. Next question. Yeah, what are the characteristics of meristematic tissues? They are living cells. They are isodiametric in shape and they are structurally and functionally undifferentiated. They have the ability to multiply. They have a central nucleus and they have a dense cytoplasm. Right. Next question.
What are the three overlapping zones of meristems?
So in medicine, there are three overlapping zones, right? Now, if you consider root apical meristem, the structure of the root apical meristem. So here you have the root and the root apical meristem is somewhere here. Surrounding that, you have the root cap, right? And yeah, it's look like this. It looks like this. Okay, I'm not good at drawing. Yeah, looks like this. And there are root hairs as well protruding like this. So there are three zones, and these three zones are overlapping. Okay, you can't specifically say this is one zone, this is the second zone, and this is the third zone because they are overlapping on one another. So this is going to be the zone of cell division, and this is going to be the zone of cell elongation, and this is going to be the zone of cell differentiation. So can you see at these points they are overlapping on one another. So that's why you can't differentiate this. You can't differentiate that this is one zone, this is another zone, and that's the third zone because they are overlapping on one another. So the three zones are zone of cell division, zone of cell elongation, and the zone of cell differentiation.
What is known as the primary growth of the root?
Yeah, what is known as the primary growth of the root? That is once again a definition. Ali.
Elongation of the root due to the primary resistance located to the root.
Yeah, elongation of the root due to the activity of the meristem that are located at the root apex is what you call as primary growth of the root. Good. Next one.
What happens to the cells produced outwards to the root apical meristem and what is the importance of its structure?
Right. The root apical meristem has the ability to produce cells both inwards and outwards. Okay. The cells that are produced inwards then move into the zone of cell elongation. In the zone of cell elongation, it elongates at least 10 times the initial length. And then it moves on to the zone of cell differentiation and it differentiates to perform a specific function. That is the fate of the cells that are produced inwards. The cells that are produced outwards, they form the structure called the root cap. They form the structure called the root cap. Root cap is like a protection that is provided to the root apical meristem. So how does actually this root cap provides protection? Against what? Friction created by the soil when the root grows through the soil. So what happens is when you think about this scenario physiologically, Now, in this structure, what happens is now the cells that are produced inwards are elongating in this area. When it elongates, what happens is this root will be pushed through the soil. It will be pushed more and more through the soil because this part elongates, this part will be pushed downwards. So when it is pushed downwards and when it travels through the soil, there is a friction that is generated from the soil on the root. So in order to provide the root apical meristem from that friction, the root cavity is removed. They can ask this in a structure, it's possible. Okay. Next one.
Mention two differences between shoot apex and root apex.
Okay. Two differences between shoot apex and root apex. I think it's easy. So when they ask such a thing in a structured essay paper, you can write as two sentences as two comparison, or else you can basically draw a very small table there as well, depending on the space they have provided. Okay. So in the shoot apex and the root apex, the shoot apex is present at the shoot tip, and the root apex is found at the root tip. That is one basic difference. Apart from that, we can say the shoot apex is the shoot apical meristem. It produces cells only inwards. Okay, it produces cells only inwards. But when it comes to the root apical meristem, it produces cells both inwards as well as outwards. The other thing is regarding the protection. The root apical meristem is protected by the root cap. The shoot apical meristem is protected by leaf primordial. Leaf primordial. When it comes to the apical, shoot apical meristem is a dome-shaped mass of cells that looks like this. Okay, and on either side of this dome-shaped mass of cells, you have two structures which arise like this, which is called a leaf primordial. So leaf primordial provides protection to the shoot apical meristem.
Mention two features in parenchyma tissue that differs from parenchyma tissue.
Right. So when such a question appears, you don't have to write the features of parenchyma tissue because they are asking two features in collenchyma tissue that is obviously different from parenchyma. Even though it's a comparison, you need to understand the wording of the question. There you don't have to write it as a comparison. Now, for example, some people might write it, the cells in collenchyma is elongated, while the parenchyma is spherical. It's possible to write like that. But that's not what they are asking. What they are asking is a feature of collenchyma that is different from parenchyma. So you don't have to write about the parenchyma there. If you write about the collenchyma, that is enough. Okay, because they are not expecting a comparison through that. Alright. So what are the features in collenchyma that differs from parenchyma? One is the cells are elongated and the cell wall is unevenly thickened. You can say these two. These are the main differences that you can see, right, compared to that of the parenchyma.
What are the types of sclerenchyma cells and about their structure?
Right. What are the two types of sclerenchyma? The sclerenchyma, sclerics and the sclerenchyma fibers, right? Sclerics and sclerenchyma fibers. So what is the structure? How is the structure of the sclerate? It is shorter, wider, has an irregular structure. That's what you have to write. It's shorter, wider, and has an irregular structure. What about the fibers? They are long, they are slender, and tapered. Now you all have completed this. So what is slender? What is tapered? What's the meaning? What is slender? What is tapered? What is tapered? Slender means it's kind of thin. That's what you want to slender. And what is tapered? Tell me another cell which has a tapered structure. that you have already studied in plants? Tracheids. Yeah. Tapered in the sense, and we have it is slender and it has tapered ends. I don't know how to do it in finger. This is how it looks like. It has tapered ends. This is how tracheids look like as well. Long, slender, with tapered ends. Right? Now, what is the next question? What is the question?
What is the importance of having secondary cell?
In xylem.
Thickening by lignin in vessel elements and tracheids.
So what is the importance? Now you know when it comes to the xylem tissue, water is transported from the bottom to the top, which means against the gravity. So when water is transported against the gravity under the negative pressure, okay, because like you will understand the negative pressure scenario when you study about the ascent of the xylem stuff, but now you will not understand. What happens is it's like water is pulled from the bottom to the top. It happens under huge pressure. Okay, it happens under huge pressure. So there is a possibility that these vessels which transport water, the vessel elements and the tracheids can collapse, can break down under the tension or under the pressure that is created, under the tension, the pull, because water is pulled from bottom to the top. So when it is pulled, there is a tension that is created. So due to this tension, there is a possibility that the walls of the cells can collapse. So to prevent the collapsing under the tension of water, the walls of vessel elements, the tracheids, are thickened by lignin, because lignin provides a very huge support. Because when lignin is deposited on a certain cell wall, that cell becomes dead. The cell is no longer viable, which means when the cell wall is deposited with lignin, it does not allow the materials to cross through it. When it does not allow the materials to cross through it or go out, what happens is the plasma membrane becomes dead. When the plasma membrane is dead, the entire plant structure is dead. Okay, that's why xylem vessel elements and tracheids are not live structures. They are considered to be dead cells. You know that, right? In xylem tissue, free cells are dead structures. One structure is a living structure. Do you know that? No? In xylem tissue, what are the four types of cells? Talk loud. Talk loud. Tracheids, okay. Xylem is elements. What else? Fiber and parenchyma. Out of these four, what is living and what are non-living? Sanjana. What is living and what is non-living? It's taught in all of us as well, as far as I remember. What is living and what is non-living? Nangi at the back, I can't remember your name. What's living and what's non-living? A very basic question. See, most of you don't study now. I can't remember your name. What are the living and non-living structures? Except parenchymal fibers are living. Except parenchymal fibers? Except parenchymal, xylem parenchyma. Fibers are non-living, okay? Let it be the phloem or the xylem. In both phloem tissue and the xylem tissue, you have fibers. Fibers are dead. Okay? So fibers, xylem vessel elements, and tracheids are dead structures, and parenchyma is a living structure. In the phloem tissue, what are the four types of cells? Yeah. Didn't study. Study. No? Vessel elements in a xylem. Total question. Hmm? phloem parenchyma having further up here. Yeah, but sieve tube elements and companion cells. So there are 4 types of cells in phloem tissue: phloem parenchyma, phloem fibers, sieve tube elements and companion cells. Out of these, what are living, what are non-living? What are living and what are non-living? No idea. Something, what are living and what are non-living? What are living and non-living? You?
Elements and cells and human pattern are being.
Yeah.
And fibers are the dead ones.
Yeah, good. Phloem tissue, 3 are living. What are the 3 sieve tube element confining cells? And parenchyma, they are living and fibers are non-living. Good. A very basic thing. Please try to study. What's the next question?
What is the structure which connects the sieve tube cells and companions?
Sieve tube elements and confining cells. I think I said it like that. All right. Now, in the phloem tissue, we have 4 different types of cells. But tell me, in all vascular plants, do we really have 4 different types of cells in the phloem tissue?
What are sieve cells and which type of cells consist of sieve?
Okay. So, yeah, as I was discussing, in the phloem tissue, in all the vascular plants, do we have all the 4 cells? No. They don't have. They don't have. When it comes to vascular plants, in all the vascular plants, do you have all 4 types of cells in the phloem tissue? Vascular plant vascular plant check. Do you have all the 4 xylem? Yeah, all the 4 cells. Now, for example, given you have studied all the different types of plants, right? Vascular plants, non-vascular plants. All right. So in that, okay, how do we separate plants as vascular plants and non-vascular plants? What's the basis of separating plants as vascular and non-vascular? Huh? Yeah, presence or absence of extensive vascular system. If the Farade extensive vascular system simply have xylem phloem. Presence or absence of xylem and phloem, according to that, we divide as vascular plants and non-vascular plants. In vascular plants, we have xylem and phloem. Right? In vascular plants, we can categorize them further into 2. What is the classification? Seeded plants and seedless vascular plants. Seedless, seeded vascular plants. Seedless vascular plants, there are 2 phyla. What are the 2 phyla of the seedless vascular plants? We have not studied it in 3 years. What are the 2 types? High school and yeah, I did the first only what are the 2 types of phyla and the seedless vascular plants?
So seedless.
Seedless. Samjha. Phylum Pterophyta, phylum Lycophyta. Pterophyta, Lycophyta. Next question you are not answering, Maria. What are the 2 classifications under phylum Lycophyta? What are the 2 groups of plants under phylum Lycophyta? Clubmosis and spikemosis. We're going to do clubmosis and spikemosis. That's what you said, right? Lycophyta can be classified into 2 as clubmosis and spikemosis. Clubmosis and spikemosis. What is the example of phylum Pterophyta? What's the example? Let me know. This example. Huh? Coconut is a non-vascular plant. What's the classification? Sorry, what's the example? Kandy? Have you heard of something called the nephrolepis? Nephrolepis is the example for this. Then under Lycophyta, what is the example for clubmosis? Lycopodium. Spikenosis? Selaginella. Okay, I'm not going to go further. I have a viral depression or multiple. So you have to sign. So I was talking about the phloem tissue. Now, as we discussed, the phloem tissue has 4 different types of cells, right? Seed tube element here, companion cells here, phloem parenchyma here, phloem fibers here, right? But all these 4 types of cells are present in the phloem tissue only in angiosperms. Only in angiosperms. Do you know what are angiosperms? Angiosperms are basically the flowering plants. Okay, in angiosperms. So, except for angiosperms, the gymnosperms and the seedless vascular plants, all these have a phloem tissue which consists of 3 types of cells. Once again, I repeat it: the gymnosperms and the seedless vascular plants. Then we have to think of seedless vascular plants can be categorized into 2 as one is angiosperms, one is gymnosperms. Vascular plants we categorized as seedless and seedless vascular plants. So, out of all these, the main category with the phloem tissue here because they are vascular plants. Except for angiosperms, all these they contain only 3 types of cells in the phloem tissue. Only in angiosperms you have all the 4 different types of cells: seed tube element, companion cell, parenchyma, and fibers. In these seedless vascular plants and gymnosperms, you cannot find seed tube element and companion cell. In seedless vascular plants and in gymnosperms, you cannot find seed tube elements and companion cells. So instead of those 2, you have one cell called the seed cell. You have one cell called the seed cell. This is a long, narrow cell. So seed cells are found in seedless vascular plants and gymnosperms. Only in the phloem of angiosperms you have all the 4 types of cells in the phloem tissue. Is that clear? Clear? That's the question. I forgot the question also. Yeah, seed cells. Seed cells are the cells that are found in seedless vascular plants and in gymnosperms. They are long, narrow cells. They are present instead of the seed tube element and the companion cell. If you have written that idea, then your answer is correct.
What is the structure which connects the seed tube cells and proteins?
Right, now when it comes to the phloem of the angiosperms, in the phloem of the angiosperms, you have all the 4 types of cells. Now, when it comes to the seed tube element, the seed tube element lacks many structures. You have learned the structure of seed tube element now, right? It lacks many structures. Why does it lack many structures? What's the reason? Even the cytoplasm of seed tube element is reduced to the periphery, which means the cytoplasm is found in the very corners. And it does not have nucleus, it does not have ribosomes, it does not have vacuoles. What's the reason for this?
To transport the sucrose.
Yeah, to transport sucrose effectively, to allow more space to transport sucrose. That's the purpose. It has kept a large lumen and reducing all the things to the periphery. Now, this seed tube element is involved in the transportation. But you know, in order to signal something, in order to give a command to a cell, in order to give a command to a tissue, you require a nucleus. So who is going to give the commands to this sieve tube element? That is done by the companion cell. So each sieve tube element, along with each sieve tube element, you have a companion cell. This companion cell, it contains the nucleus, the ribosome, the mitochondria, and everything. So the nucleus in the companion cell acts as the manager to control the sieve tube element as well. The activities of the sieve tube element is controlled or managed by the nucleus in the companion cell. Okay. Again, we are the commands then. So each sieve tube element has a companion cell along with it. Fair enough. Is it clear? Not clear. Right. Now these two structures, okay? These two structures are interconnected by a special type of a structure. These two structures are interconnected by a special structure called plasmodesmata. Plasmodesmata. Not only one plasmodesmata, there are numerous plasmodesmata which connects the sieve tube element with the companion cell. Okay. Plasmodesmata is not what are plasmodesmata? They are the living connection in the plants, right? One type of we can say it is a specialized type of cell junction. What are cell junctions? What are cell junctions? This is called integrated learning. If you don't have integrated learning, you can't pass the exam. So tell me what are cell junctions? What are cell junctions? Huh? I didn't hear you. They are not checked by you're talking about the cell side. What are cell junctions? Definitions are definitions memorize karana. The structures at which the cytoplasm of the adjoining cells join. The structures at which the cytoplasm of adjoining cells join. Okay? Guys, just started unit 4. Unit 1 make it just 2 units in biology. What am I doing? Okay? Yeah. Yeah. So the companion cells and the sieve tube elements, they are tightly interconnected by numerous plasmodesmata. That's the question, right? Which structure does this? Next one. This stuff we learned today. Which structure blocks the cortical apoplast from the vascular apoplast? I know you don't know what is apoplast, but I said you what is cortical apoplast and what is vascular apoplast, isn't it? So, what is the structure which blocks both? The correct answer is not endodermis. The endodermis doesn't block the Casparian strip or the suberin belt of the endodermis is what blocks the cortical apoplast from the vascular apoplast. Clear?
What are the two main functions of the root cortex?
What are the two main functions of the root cortex? I think I should mention when I was explaining, I mentioned only one function. One thing is it helps to transport water and minerals towards the vascular cylinder because then if I draw it here, you have the epidermis like this, and then you have the endodermis like this. So here there are root hairs. The water that is absorbed through the root hairs is transported towards the vascular cylinder through the cortex. That is one function of the cortex. The water and minerals are transported towards the vascular cylinder. What is the other function? The other function is cortex has the ability to store carbohydrates. Why? Once again, integration. Cortex is mainly composed of parenchyma cells. Because it is mainly composed of parenchyma cells, can you remember? You might have studied in the last week class. Parenchyma cells has one function called storage. Can you remember? Yeah, it can store carbohydrates as well. Is that clear? Clear? Answer the question.
Draw and label the primary structures of the primary monocot and diamonds.
And if you haven't got that, Dicot step. Again. Come, the word. Come. Okay. Come the word. And you can also take your friend if you like. Come, both of you. See. Fine. She's okay. Why are you not well? Is it hard to be on this? Shall I switch off? Yeah. Oh, I already different problem. Okay, so you go. And then come. I'm going to give you the monocot step, not the dicot step. Easy one, come and sit. Come. Right, quickly. Hey, don't waste time for me. Come. वहाँ तो आगे तक तरह के लेबर करंट में निकले तो हमारा total monocot step. Man, you go and label it. So you have to label the round tissue, the epidermis, and the vascular. Will you do that? Others look through the other structure. Down there. Why did you do this stuff? Why did you do this? What are these? These are also vascular bundles, but you should have been scattered. We will go to as well. And come and label. Please, I'm trying to make you active in the class. No, couple of everything. Come with you. Quickly. Okay, so it's consuming time. This is the epidermis. And then these are the vascular bundles. You haven't drawn the sclerotype, cluster of sclerotype. When you draw the vascular bundles, at least in one show the cluster of sclerotype. And then mark this as the vascular bundle when you are labeling, and then mark this, label this as the cluster of sclerotype cells. And this as the ground tissue, because the ground tissue in monocot stems is not differentiated into cortex and pith. Okay, right. Dicot stem. I'll erase this. Sanjana, come to label it. No, so. Okay. Now, often she labels, I'll tell you, make it more accurate. I find, no? You can ask the question first. Thank you. Right. So when you know the structure of the Dicot stem, one thing is this is correct, not wrong. But still, when you're drawing the structure of this is the epidermis, has she marked that? Yes, epidermis that. In order to the epidermis, we have the ground tissue which is differentiated into the cortex and the fin. So here, there's no prominent cortex, the way you have drawn. Okay, can you see the cortex is a thin layer? So you have to allow more space to the cortex and draw it kind of constricted because pith is a small area compared to the cortex. So if you draw improportional in your exam, you won't get marks. Okay, they feel like you have no idea about it. So draw it proportionally. So make it, a bit constricted. And then this area is the cortex. Yes, marked. This area is the cortex and this is the pith. And in the vascular bundle, facing the pith, you have the phloem. Facing the pith, you have the xylem, not the phloem. Inner structure is the xylem. Outer structure is the phloem, isn't it? Okay, so this should be interchanged. Okay, this is phloem. Phloem is the structure which is facing the cortex, and xylem is the structure which is facing the pith. And surrounding the vascular bundles, you have a cluster of sclerenchyma cells. Okay, and because this is a dicot stem, in between the xylem and the phloem, you also have to mention about the vascular Kandy. Okay. Clear? Proszę wiem, proszę. Tak, wiem. Everything's clear. Okay.
Mention two differences between Dicot and Monocot stems.
Okay, they can ask this question also. Mention two differences between dicot and monocot stem. One thing is, in dicot stem, ground tissue is differentiated into cortex and pith. In monocot stem, the ground tissue is not differentiated. Okay. When it comes to the vascular bundles, in dicot stem, the vascular bundles are arranged in a ring. But in monocot stem, the vascular bundles are scattered throughout the ground tissue. There's no proper arrangement. And the other difference is, here the vascular bundle contains a vascular cambium. In monocots, they do not have a vascular cambium because it does not undergo secondary growth. Okay? Those are the three differences you can write into. And yeah. Okay. Online students, any doubts? Next, we have to start off with secondary growth. But the thing is, I'm not planning to start the secondary growth. We start the secondary growth. It's going to take at least 2 hours for me not to explain, but for you to understand. So I'm skipping that part for the other day. So I'm going to start the shoot architecture and the line patch, which is 121st page. Okay. Shoot architecture and the light capture. Page number 121. Right. Now, when it comes to plants, okay, this is a essay section. When it comes to plants require light to carry out photosynthesis. So, suppose you have a forest in a forest. There are a lot of plants. There are tall plants, there are short plants, there are intermediate height plants, and yeah, it's a very canopy. It's basically there are layers of plants. So usually when sunlight falls, the most of the sunlight is absorbed by the tall plants. So what happens is even the short plants are in need of light. So in order to search light, it always tries to increase its height or maybe change its branching pattern, maybe change its leaf pattern or do something to capture the maximum amount of light. So always in nature, the plants, they do arrange themselves in order to obtain the maximum amount of light without damaging the chlorophylls, because over intensity, anything too much is not good. Okay, so over intensity of light too can damage the chlorophylls. So to capture the maximum amount of light that is required by the plants can do different adaptations. It can do adaptations in its architecture. It can change architecture, this is basically its structure. Okay, so there are, now for example, when it comes to a plant, in plants can change three things in its architecture in order to obtain the maximum amount of light. Okay, one is the stem. One thing is the stem. The other one is regarding the leaves. The other one is regarding the branching pattern. Branching pattern. Okay. So these are the factors which decides how the plants capture the maximum amount of light. Okay. Now, for example, if you consider a structure, I mean a forest environment. In a forest environment, because these plants receive the maximum amount of light, the woody plants, those are showed, they allocate more of its resources, more of its growth substances for them to grow even more taller to capture more and more light. Okay, that's how the stem adapts its architecture. The stem, it grows taller and taller. Now, for example, if you take a vine, but you know what are vines, right? Vine can well, can see that vines. I think it haven't shaped. Do you know the time? Do you know Sika? Vines. What's the time word? What's the time word? Well done, vine. Do you know? Gosh, what is it? Remember? Vines. Can you please search and tell me the meaning? Time word. What are vines? Huh? Khodi. Khodi. Okay, whatever. Okay, well done, vine. Now, when it comes to those vines, they don't have a strong stem. You understand, right? They don't have a strong stem. So, can they grow, against the gravity? Can they stand against the gravity? No, because their stem doesn't have a strong mechanical support. So, they always try to, stick on to some other person. Maybe it can be a runner, or maybe it can, bind around another structure, go up. So, basically, vines, in order to capture the maximum amount of light, they rely on another structure and then they grow through the structure to reach the maximum amount of light. So, in order to capture the maximum amount of light, we have three adaptations that can happen. One adaptation can happen in the stem. Okay, one adaptation can happen in the stem. In the stem, what happens is when it does not receive enough light, it allocates more of its resources to let the stem grow taller to capture the maximum amount of light. Okay, and when it comes to plants like vines cannot grow tall, it cannot grow by itself because it does not have a strong mechanical support. So it relies on some other object to reach the maximum amount of light. Is it clear? Once again, we'll read through the book and cover. Okay. So in the book it says, length of the stem, the branching pattern are designed to capture the maximum amount of light. Plants grow tall to avoid shading from nearby plants. That means these plants, they allocate more of its resources to grow taller. Why? Because this provides a certain shade. Shading in the sense, does it receive enough light? No. So because it does not receive enough light, in order to avoid that, this plant tends to grow tall. Is that clear? Clear? If you have any doubts, if you want me to explain again, tell me. Okay? Sure? Yeah. Right. When it comes to the stem, most tall plants have a thick stem with strong mechanical support. Now, for example, a mango tree, they have a thick stem. That's basic, right? When the plants grow further taller and taller, why does secondary growth happen? In order to provide more and more mechanical support. Because sometimes when the plant grows taller and taller further, there will be a certain imbalance. So in order to provide the proper balance, only there is a very thick stem at the bottom. Woody plants undergo secondary growth, thereby making their tall stem stronger. Vines rely on other objects to reach higher levels to capture more light. Okay, so that's regarding stem. Very basic. It can be made complicated with the in order to avoid shading, the stems are allocating more fixed resources to grow tall. Okay, when it grows taller, what happens is the tendency of receiving light will increase. What happens is it receives enough light. Okay, and what happens is vines cannot stand by itself, so it relies on other objects to grow higher and receive more light. Okay, so when it grows taller, in order to prevent the imbalance that is happening, what happens is the plants they widen their stem. Basically, most of the plants like dicots, they undergo secondary growth to make their tall stem stronger by making its diameter wide. Okay, that's what is happening in stem. And then regarding the branching pattern, now you know there are unbranched stems as well as branched stems. Now, when it comes to most of the monocots have unbranched stems. So when they are unbranched, the tendency of receiving light is kind of relatively low compared to branched stem. The coconut tree is very tall. So it receives enough light and also the leaf size is very small. In the coconut, the leaf size, the coconut leaves, it has a very small area. So the light that is received by that, even having an unbranched stem, is enough for them to carry out photosynthesis. But when it comes to a tree like mango, it has a very good branching pattern. So this branching allows the leaves to receive enough light to carry out photosynthesis. So this branched nature also satisfies the required amount of light by the plants. Is that clear? No? No, sure. The other one is regarding the leaves. Okay. Of the leaves we can talk about them under three structures, okay, or three factors. Now, when we talk about the leaves can vary in their size. Okay. Leaves can vary in their phyllotaxy. I'll tell you what phyllotaxy is. Also, leaves can vary in their orientation. Leaf variations there are under three factors. Now, we spoke about the variations that occur in the stem. We spoke about the variations that occur in the branching pattern, the branched, unbranched nature, isn't it? Also, under the leaves, the variations that can occur according to the leaf size, the phyllotaxy, and the orientation. Okay. When it comes to the leaf size, we have larger leaves and also we have smaller leaves. Large and smaller of the large surface area and small surface area. Okay. Now, larger leaves are found in, or broader leaves are found in which kind of environments? Large leaves are found in which kind of environments? Especially in rainforests. The reason is in rainforest, I don't know whether you have been to any, I've been to Singharaja. So in rainforest, the amount of light that is received by the lower, lower grade plants, the low canopy plants are very low. At the same time, these rainforests, they don't get enough sunlight throughout the year. Those areas, they don't get enough sunlight throughout the year. So these broad leaves are found in those kind of areas like rainforest, so that broader leaf area means broader surface area. Broader surface area can capture more light. It can capture more light. When it's broader, it can the amount of light absorbed by this is greater than the amount of light absorbed by this. Obvious. Because the surface area of this is very low. Surface area of this is very high. So the light absorbed here is very high because the light amount received by this environment is low. In rainforest, the amount of light, the amount of sunlight is low. When there is sunlight, it tries to absorb a lot. Maximum you understand? So in rainforest, they have a broad surface area. The leaves they have a broad surface area which absorbs a maximum amount of light. But in very dry or very cold environments, the leaf surface area is low. The reason is actually to prevent transpiration. Transpiration is another thing. Have you heard of transpiration? You have learned about transpiration. You haven't learned the procedure, but you have heard this word and the unit 2 when you studied about the water. Okay, ability to moderate temperature, right? So during transpiration, actually what happens is water is lost as water vapor from the plant body. It mainly happens through the leaves. Okay, so the main avenues of water loss in plants is the leaves. So what happens is in very dry or very cold environments, the humidity, the amount of water vapor in the environment is low. Very dry and a very cold environment. The amount of water vapor in the atmosphere, I'm not talking about the leaf, in the atmosphere is low. When it is low, what happens is always water vapor tries to escape from the plant through the leaf. Atmosphere plant because it is supplying, it is getting water supply from the roots, through the soil cell. So there is an imbalance. Can you see? In atmosphere and then in the leaf it's really high, in the plant it's really high. So therefore always what happens is in order to create a balance, Vatura plant again the water vapor in the water. Water vapor always tries to come out of the plant body to increase the water vapor amount in the atmosphere. That's what's happening. So that water loss happens through these leaves. When the surface area of the leaf is higher, the water loss becomes higher. Stomata, the stomata, amount of stomata is high. So the water loss through the stomata is high. So that's why in very dry or very cold environments, they have very thin leaves. The surface area of the leaves is very thin. So that they can also reduce transpiration, and that is enough for them to capture the amount of light they require. Okay, this is a variation that happens in leaf size. So leaf size, they can be broader or they can be smaller like this. Broader leaves are found in the rainforest. Smaller ones are found in very dry or very cold environments. Is that clear? The other one is the phyllotaxy. Phyllotaxy in the sense, the arrangement of leaves around a leaf node. If you take this as a stem, and if you take this as one node from which the leaf arises, the number of leaves that are arranged per node. In some plants, you might have seen there's only one like this may make it as a material. But in some plants, per node, there can be around 3 leaves like this. Have you seen this kind of plants? Obviously, yes. And then in certain plants, what happens is there are multiple leaves per node. There are multiple leaves per node. So this branching pattern is also a variation which determines how the light capture occurs in plants. Okay, so according to the requirements of different types of plants, the phyllotaxy changes. Then one leaf per node theory, the efficiency of light absorption is high. One leaf per node, so it has more space. Now, in these, the efficiency of light absorption is kind of relatively low. Even the area, the size of the leaf is low enough in these kind of plant arrangement. So that is adequate for the plant. This is adequate for this plant. So phyllotaxy is another variation under the leaves which determines how the plants capture the light. Okay, phyllotaxy is the arrangement of the leaves around a node, or else arrangement of leaves in the stem, we could say. Okay, it can be 2 leaves per node, it can be 1 leaf per node, it can be several leaves per node, likewise. The final one is the leaf orientation. Leaf orientation in the sense whether the leaves are placed vertically or horizontally. That means when the light is falling like this from the sun, whether the leaves are placed horizontally or whether the leaves are placed vertically like this. Now in grasses and all, the leaves are placed vertical, almost vertical, 100% vertical. It's almost vertical. This is also not 90 degrees or 180 degrees. It's almost horizontal. Okay, nothing in nature is perfect, right? So this is almost horizontal, this is almost vertical. So this leaf orientation is also because of the requirements of the light by the plant. Now, for example, leaf orientation is vertical in plants like grasses because that a vertical leaf orientation is required to prevent the damage that is caused by the overexposure to light. Now what happens is, when the leaf orientation is vertical, the light rays are falling parallel to it. When the light rays are falling parallel to it, the tendency of absorption is a bit low. When it is falling perpendicular to the leaf, the absorption is high. So in a horizontal leaf, the horizontal orientation, the light absorption is high, and in a vertical orientation, the light absorption is low because the light rays are falling parallel to the leaf. Is that clear? Right. And these kind of leaves, the horizontal leaves, are important in low light conditions. When the light is low, the supply of light is low, when the receiving amount of light is low, when the amount of light received is low, when it is horizontal, it can capture the maximum amount of light even in low light conditions because it's horizontally placed. All the light rays are falling perpendicular. When it comes to the vertical orientation, the vertical orientation is important in an over-intensive light. When the light supply is very high, when the intensity of the light is very high, when the intensity of the light is very high, it can cause damage to the chlorophyll sometimes. So to prevent damage, the leaf orientation is vertical. Okay. Is it clear? Right, go through the in the resource book, please. No. How far is it? Okay. Okay. Is that clear? Any doubts? No. Ali, is that clear? Sure. Ali, clear? Sure. Prashna. Yeah. I clear. So yeah, that's it for the day. It's almost 11:10. I have to that's very much for you. Yeah, anything that you want to say, like, any criticisms? Criticisms? Seems like Avi has a lot of criticisms. You can tell me anything. Are you all satisfied at least a little? 50%? Did you all understand today's lesson? Exactly, sure. You can drop me a message anytime by coverage 076 418 428. 076 418 428. All right, then. So yeah, y'all can leave. Online students, y'all can leave as well. Thank you for joining. And and not great something is not good as I will study. So I would like to study. Procrastinating. How to call it? Now, because you are enough that you are pregnant for school life. Okay, school life. We go to school every day. Alright. So what I did was that I targeted to cover a certain number of pages in a day. So, for example, we take the biology, there are around 70, 60 pages, around 60 pages. So, I'll target to cover up 12 pages every day. And for once, which I can take around 5 to 10 minutes. So, to memorize the whole thing, something very, yeah, to memorize the whole thing. So likewise, that's how I used to cover. And the main thing is, you should not study biology just 2 or 3 days a week, at least 6 days a week. That's what I would say. Now, if you start at least from 1 hour per day for biology, that's more than enough enough. Now, I'll give you a small task. By next week, before the next class, or you're coming to the class before the next class, you have to cover at least in unit 2, at least until enzymes, which is not much. Like until the definition of enzymes. Okay, so 6 days is more than enough for them. And also, like I gave you some questions, and also the section that we did, the section we did was very small because we had to revise all the sheets along, right? So you can revise that as well. So that's how you start. The thing is, you have to be consistent. Now, when you study this week, maybe in a week you have to revise it quickly from A to Z. That's how you do it. You try that and let me know. You can drop me a message also.
But can I have that book celebrate next week?
Oh, my book. This one, right? Probably.
I already uploaded said I did basic development for biomass.
You?
Yeah.
What's your name? What's your name?
Decided.
Can you drop me a message again? I think I haven't saved.
I didn't say.
Yeah. Please drop me a message.
Bye.
Bye. How's it going? Good.
Nice.
No. Problem.
You should have it done.
Thanks. So you have any class couples? Did you know? Did you know?
For this, they can stay exam at the I will say.
Well, I'm working on it. You can focus on this.
Catch up close. Work this in months and.
Course is now actually recording. Call. Right? Here,
I must think twice. There go one option.
watching that study because there are not like to study but the basics take on. There you go, yeah, allows them to do fast. Anyways, from next Thursday, not a study from will be studying it. Then we can join weekdays also who was regular from yeah. Bye.
Nice. Yeah, yes, so.
Thank you. Oh, that is a video. Bye. Yeah. That's it.
How to study physics and chemistry?
Chemistry, who will go?
Nothing this way.
Oh, man. It's just
You gotta finish third assignment and moving to lectures.
studio regular first 5 days are 10% of the syllabus. So thing is this now you have guessed
Yeah.
in phase 2.
Huh?